Welcome to video 1 of Genetics, the Code Broken. In this video, we're going to be looking at DNA and polypeptide synthesis, some of which we've covered in our previous topics, but we're going into a little bit more detail in this option topic. So we're going to look at how DNA allows us to have the information to produce RNA, which allows us to then produce a polypeptide. You're going to be creating a model of DNA in class, and we're also going to be looking at gene expression, which will form your research task in class, but we'll cover a bit of it in this video today. So remember, when we talk about DNA, we are in fact talking about deoxyribonucleic acid. And the reason it's called deoxyribose is because it lacks an oxygen. So RNA does have that oxygen in that sugar molecule. Remember that when we talk about a nucleotide, we're talking about a sugar, a phosphate, and a base. And a nitrogenous base, we have 4, A, T, G, and C. And remember that in RNA, the thymine is not produced in mRNA, so we do have uracil. A gene, remember, is a region of DNA that controls an inherited characteristic, and that's by controlling protein synthesis. The structure of DNA is so that it allows us to have a code which gives us the information to produce the polypeptides. And if you don't know what a polypeptide is, I've said it already 5 times, but it is a sequence of a chain of amino acids that form the basic primary structure of a protein. Remember that the information in the DNA codes for these amino acids that make up the polypeptide. There are in fact 20 amino acids in humans, and they're coded by a triplet of bases called a codon. A codon determines which amino acids are going to be incorporated into the polypeptide, the number of amino acids, and the arrangement of these amino acids in the polypeptide. Most of the DNA in chromosomes, in fact, does not code for RNA molecules or proteins, but is essential for the cell and organism. And we're going to look at those later, and we're going to look at how gene expression relates to these structures. Remember that I don't have it in the video, but there is a table which has all the codons and the series of 3 letters which code for each of those amino acids. So when we look at polypeptide synthesis, we are in fact looking at 4 processes which work together to produce that polypeptide. We have looked at transcription and translation before, but 2 new processes are regulation and RNA splicing, and these occur in between transcription and translation to make up the 4 processes we are going to cover. So DNA transcription, or the copying phase, occurs within the nucleus, and DNA strands unwind because of the DNA helicase molecule, and it becomes a template on which a complementary copy of that code is built. This copy is in fact an RNA molecule called messenger RNA, or mRNA for short. So when it is unwound, the RNA polymerase moves along the DNA and creates a short double-stranded DNA RNA molecule. And as mRNA nucleotides are joined together, the DNA helicase reforms just behind it and releases the developing single-stranded mRNA. We're going to be looking at an operon on the next slide, and that incorporates an operator gene, a promoter, and 2 or more structural genes. And this operon is important for producing an mRNA molecule. When we talk about regulation, we are talking about where regulator genes produce regulatory proteins, and these affect the activity of other genes. So when these regulator genes binds to the operator genes, and they can control which region of the DNA is copied, the rate of that transcription, and how many RNA transcripts are made from the gene. Remember that the first and last codons in the gene act as switches that turn the transcription process on and off, and they are in fact coded by the amino acid, by those codons, the start and stop codons, and this regulates the length of the mRNA that is formed. So there is a region at the start of the gene, which is called a gene promoter, which ensures that the RNA polymerase binds to that DNA strand and that the gene is copied in the correct direction. The third process is called RNA splicing, and this occurs again in the cell nucleus. So in our RNA molecule, there are what we call exons and introns. And exons are coding sequences, so they contain the codes for amino acids. And introns are non-coding sequences, so they don't actually code for any amino acids, so therefore they're not important in the production of a polypeptide. So the whole gene is transcribed into that mRNA molecule in that transcription phase, but then the introns are cut out. And this provides a way of rapidly producing many variations on the original protein. And that large mRNA molecule can be spliced in a number of different ways to create different messages, and therefore producing many different protein products from the one gene. So here we can see we've got our DNA, it's transcripted. We see that we do have the RNA molecule with intron and exon sequences, and then we get our mRNA molecule that has only exon sequences remaining. And then we have our fourth and final phase, which is translation, and that decodes the mRNA to our amino acids and therefore to our polypeptide. So unlike the other three processes, this occurs outside the nucleus at the ribosomes. So transfer RNA molecules, or tRNA for short, in the cytoplasm, and they each carry a specific amino acid. Every amino acid has at least one transfer RNA assigned to carry it, but some amino acids have many more than that. So we can almost think of tRNA is in the middle. On one end of that tRNA molecule, mRNA molecule binds to it, and the other end binds to its amino acid. So during this translation process, all the amino acids are brought together in the correct sequence by those tRNA molecules that are following the mRNA code. And this activates the amino acids so that they can form peptide bonds with the adjacent amino acids, thereby producing a polypeptide. So I've seen this diagram before. Remember, we go the DNA template, we produce our mRNA, and then we translate it into our polypeptide. So when we talk about gene expression, we are talking about the information that's in the DNA code that is being used to make a polypeptide by undergoing transcription and translation. And these create the observable features in a cell or organism. So there are controls for gene expression at the transcription step mainly, but there can be other controls that alter the mRNA and what it does and whether that those genes are expressed in a cell or an organism. So here we have an image of 6 control points which can control gene expression. The first is the control of transcription, and that can control when and how genes are transcribed. Then there's the control of the mRNA molecule. So this can control where splicing occurs, and therefore different splices can produce different polypeptides. We can control the mRNA molecules that pass into the cytoplasm for translation. We can control the molecules that are selected for translation. There are sometimes mRNA molecules that are no longer required. And then the last one, we can control the proteins that are produced through gene expression. One specific way which gene expression is regulated is by regulator genes, and these produce a repressor protein that can affect the activity of genes by switching off the transcription of a structural gene. So when this occurs, it stops the action of RNA polymerase, therefore it doesn't produce the polypeptide product that it would have, but when the repressor is taken away, the RNA polymerase is able to transcribe the operon. Thank you for watching video 1. Make sure you tune in for